And you don't need to get on the internet, or do you need to about a minute or so.
Tomorrow I didn't write down.
Okay, so tomorrow I'll make sure we get our ahead of time.
Okay.
Download from the internet.
No problem.
Why don't you just log
And it's teaching demos so people can actually, if they want, they can install.
You're going to make sure that
None of these guys are up and running, but oh, so that clock is about 5 minutes slow, just for, you know. And so we're gonna go to, and there's no hard end to this, so 50 minutes, whatever, something like that, and then some questions, and then we'll have some time for one-on-one, and then we'll head out to the restaurant. So cool. Tomorrow's a little bit more of a time thing, but either way, it's not gonna go.
Yeah, because they have sort of like everything from you've never installed R before. I'll provide that stuff, and people can sort of like be working if they want to follow along. Then there's like the data, getting the data ready to sort of start the time. But I want to jump right to the plan.
Totally. Yeah, so it's all whatever. And then I think is it? I don't apologize.
That's how I feel. That's what I thought, but then I was like, wait, is that
All right, so we're gonna get going here in a sec, you guys. Right before we start, though, just to reiterate, if you guys haven't, I don't know what I know, kind of crazy schedules on Tuesdays, but we're gonna hear from Dr. Ladin tonight. There's also another opportunity to hear from him. This is Research Talk. His teaching demo will be tomorrow in the same room at 10 a.m. And another opportunity right after that for any students that want to hang out and talk questions or talk shop about stuff. Perfect opportunity for you guys to do that then. And then I just remind you guys, if you guys go to our website with his stuff, there's a question and answer form there, or not question and answer form, sorry, feedback form. So by all means, we welcome all your guys' opinions. So please do give us any feedback. And that only goes to the committee, to nobody else. So please do that. And with that, I think we're probably about on time, so we should get going. So welcome everybody here to our research presentation. Zach Ladin, Dr. Zach Ladin, is joining us from the University of Delaware. And he comes originally from Rhode Island, did his undergrad at Rhode Island, right? And then went to Delaware for his master's and PhD, and he's there as a research fellow, but he's a postdoctoral scholar, but he's also had some cool points in between doing projects with the Park Service, doing, what else? A little stint down to Singularity University at Google's campus and all that kind of fun stuff. So all kinds of neat things to be had. So a great example of someone that started on one path and still is going down that path, but has added a bunch of cool other interests and other abilities and things of that nature. So without further ado, here's ecology in the Anthropocene. All right, Zach.
Hey, well, I just want to first say thank you for having me. I'm very grateful for this opportunity to be here and to tell you about how I got here to this point and a little bit about my research. So my talk is sort of the beginning. I'm going to sort of go through very high level what really gets me going, fired up, why I'm doing what I do. And then I'll give you some examples of some research that I've been involved with. And after that, I'll hopefully have time still to go through some new, what I think are pretty exciting projects that are happening now and that are on the recent horizon, upcoming projects. And so, so we've entered a new geological age, the Anthropocene. Has everyone entered it with us? Yeah. Does anyone haven't heard that term yet? The Anthropocene? Okay, great. Well, we're having such a big impact, humans, on the planet now that we've earned our own place in geologic time, that we're now the major driver of what's going on this beautiful planet here. And if my clicker will cooperate, I like to start with this. This is, obviously it's our planet, right? But this is a view from the Himawari-8, a Japanese weather satellite. And I just want to take a step back and hopefully we can collectively have all our minds blown together. This always blows my mind. Okay, 3.4 billion years ago, life emerges on a planet, and it's gone through all these trials and tribulations, and eventually, somehow, miraculously, humans evolve. Consciousness emerges. Tool use emerges. To the extent that we're building tools that we've put out into space, orbiting our planet, we're looking back at ourselves for the first time. That just blows my mind. Every day, remembering that. We take all this for granted. All your cell phones, your GPS, everything we walk around, we eat and breathe this technology now. We forget just about how we got here and where we are and how magical this point in time is. So I always like to start there, remind myself what's going on here. It's now estimated there's potentially over 1 trillion species on the planet. Okay, you go out and sample the soil. I'm sure if you wanted to and sampled in this room, you're bound to find a new species that's been undocumented before. We know virtually nothing about our own planet. So that's also something I also want to try to remember as I'm going out there and trying to understand these systems and study these systems. That's about 1%. Okay, so we've got the vertebrates, the plants, maybe the invertebrate community, not so much. And then as soon as you go, to these microscopic organisms, bacteria, fungi, viruses, things like that, we know virtually nothing. And we're sampling more and more. We're finding new species all the time. So that's also something really impressive point I always try to make, that we need to also be aware of our naivete about what's actually out there. And that's an inspiring thing. This isn't a bad thing. This is an exciting thing. It's a time of discovery for us. We now have the technologies to go out there, and you have them in your hands. You have the power in your smartphone to be able to capture these data, look at these data, and now to go out and collect new data and add that to this collective knowledge that we're building. So a couple quotes here that really, from a guy, some of you might already know who this is from, and some might not be familiar with this, but this gentleman, when I first heard some of his words, changed my life and the trajectory of my path on this planet. But this is one of the basic tenets of his thinking. Very smart guy. If you're going to go into a system and start trying to understand it, and you take it apart and you try to understand it, it doesn't make sense to throw away the parts and then try to put it back together. I was really a curious kid. I would take apart all the electronics around my house. My parents hated it. And I was always really good about taking stuff apart, but I could never get it to go back together and have it functioning again. So that's why I'm not an engineer. But this is this idea. It's important to keep all the parts. Have we learned this yet? Okay, have we actually learned this idea of conservation? Should we conserve these parts? And we're not talking about cogs in a machine. We're talking about biodiversity. We're talking about organisms and species here. Have we learned this? No, because we as scientists don't even get it yet. We, the scientists who are out there studying this stuff, we don't even understand it. Who said this? Yeah, Aldo Leopold. If you haven't taken the time and checked out Sand County Almanac, hopefully those of you who have enjoyed that. But this is back in the 1940s, 50s. Very forward-thinking, very smart guy. So we might think we're intelligent, but are we? So as we go out and modify these landscapes, as we modify now the planet as a whole, the climate, this is the biggest challenge our species has had to face and will face ever. And it's become a survival situation. So we need to really start to question. Cities don't develop by design. They're sort of just this blind growth, like a hyphal colony spreading out into the environment. So it's very interesting. Does everyone remember Ghostbusters? It's that scene when the world's about to explode? Now, I'm just kidding. This is actually the tribute to the 9/11 towers, where we shine these giant lights up into the sky. And we do a lot of things as humans that we do. It's our social aspects, our sort of cultural aspects. And I'll just put this up as one tiny example that here we are doing something for a good cause. It's very noble to honor the people that died. But when we fire these giant lights up into the sky, when you zoom in, what we see are birds being attracted thousands and thousands of birds that get confused, disoriented on migration, because they're queuing in on stars and the moon, and to try to find their way to their breeding grounds or wintering grounds. And so everything we do has repercussions. So what I do as an ecologist, I try to use data to understand how the effects of humanity are influencing populations and individuals and communities. And this is just an example of some of the work we do is where we're tracking individuals. This is movebank.org, an amazing collection of data sets where people are posting biologging and biotracking movements of birds across continents. We're also using next-red radar to track densities of birds, bats, and insects. What we're seeing is the emergence of birds. That's actually not weather. We call that bioscatter. So we're using that in some projects now. This is amazing, crowdsourced data from eBird. Any eBirders here? Hey, awesome. So we can now, with the power of our smartphone, take it out. I can see a bird. I can put it in my location, update, send it to the cloud, and it's going to go in as a data point that can then give rise to these beautiful models. What we're seeing is here, the wood thrush, a species I study, is arriving for breeding on the breeding grounds. This is time here on the x-axis. It's the time lapse, and then they're sort of moving back down to the tropics. These birds are actually tropical species. They just take a vacation up here to breed, and then they head back home every year. We think of them as our species typically. So really, this is what I call my massively transformative purpose. This is why I do what I do. It's the fire inside me. It's what I encourage all my students to try to find, that really burning passion, because there are going to be challenges. There are going to be hard things you're going to have to work through, but none of it's hard when you have a passion that's so important to you that you're willing to just push through. This is really something I talk about all the time, and I'm really enamored with this idea that there's latent information stored in the movement of a bird or a movement of the population trend of insects or plants. And this information that's stored in these ecosystems, I think, is the most valuable thing we have. It's our most valuable asset. We talk about ecosystem services. We talk about the functioning ecosystems we need. That's why we protect areas. That's why we do conservation work. But I think the next paradigm will be the information that these systems are giving us. They're telling us how to behave, how to course correct. If we listen, if we know how to interpret the signal, if we can separate the signal from all that noise in there, and then understand what these populations are telling us, this is the most valuable predictive tool we have. And that just blows my mind, that we can build a model that predict the future. Just think about that for a second. That we can look at an ecosystem or measure a population of organisms, fit a model, and then use that to predict what's going to happen tomorrow before we even arrive there. That's amazing. That's an amazing power we have. And then everything gets couched in this context. Okay? Nothing is in isolation anymore. Everything we do now is having these huge ramifications. So we have to be incorporating our society, our behavior, our economics, our politics. All that stuff has to be talked about, has to be thought about carefully in concert with all the data we're collecting on these bioindicators, these bird populations, these insect populations that are being influenced by our activities. And so humans, we're organisms, right? We're animals also on the planet. We have these beautiful sort of manifestations of what we do. Look at, there's some beautiful examples of what we do. Just modifying the landscape for growing agriculture. There's inherent sort of mathematical beauty in the results of our behaviors. There's also a very dark side to what's happening. We're supporting a growing population. Populations grow exponentially given unlimited resources, and our technology keeps pushing that bar up, that carrying capacity. We keep opening the window on how much resources are available for our population. So we're going to experience exponential growth until we become limited by some other factors. So can we continue this sort of unbridled growth? There's papers that talk about biological annihilation, mass extinction. There's a nice dialogue going back and forth. If we were in a sixth mass extinction, some people say, well, we wouldn't be here talking right now. You'd have fundamental ecological collapse in most cases. Food systems, falling apart. E.O. Wilson's talked about if we got rid of ants, that we'd have about 2 weeks for our own human population to survive. We just got rid of all the ants on the planet. So things would be going on, maybe not quite as good as they are now, but this is definitely something that could be on the brink. And we could be feeding this. So we have to be cognizant of where we're headed. You know, everyone knows we're getting warmer. Humans are causing it. It's not a debate anymore. This is human-induced climate change. But these gentlemen still have to feed their families. Those 2 guys who are cutting down these forests in the Amazon to make way for soybeans or corn that then get turned into cow pastures, they're responsible for probably another 10 individuals that they have to feed at home. So how can we say that they're doing something wrong? You know, their families, they're trying to survive. So that's not the solution. You can't point the finger and say, no more deforestation. We need to get into the system and understand the social problems and try to correct those. And I think there are economic solutions that need to replace the current extractive systems we have today that are slash and burn, that are giving rise to these massive, more frequent, more intense storms. There's no more 100-year storms. That concept is gone now. So now we talk in decades, like, you know, every storm is a 100-year storm potentially. So we continue to see amazing devastation. Coastal systems like this that are not designed to withstand these types of storms that we encounter now. And what will happen if we just continue business as usual? Well, it's already hit the popular, you know, it's in our popular press, it's in our sort of social consciousness that, okay, you know, we're really not in a great spot here. We have to do something about it. Yet we continue. Every day, each of us, we're all a little bit responsible in leading our lives and trying to make our own ways in the world. And, you know, this can't go on forever. So that's what I do. That's what really gets me pumped up. And I think this is a really important thing to be working on. So these are just a couple shots of me in the field. I love birds, and I do some bench chemistry stuff too. Even go to the quarantine lab once in a while. So that's just me out in the field. If I were a race car driver, these are some of the patches I might have in my car. And then these are some of my sort of academic or intellectual idols I've gotten to meet over the years. I think this guy, Stephen Wolfram, on the far left, mathematical genius. He was interested why I cared about birds. And I'm like, well, birds are these bioindicators. They can help us figure out, you know, how to manage our land resources. And he's like, well, can they predict the weather for us? So even people that are so bright aren't, you know, getting it. You tell people you study birds or I count birds. They have no conception why you might want to do that. There's Ed Wilson. I was actually in an argument with him about whether biophilia exists or not. But yeah, Ed Wilson, I think his famous quote, we have Paleolithic emotions with medieval institutions and godlike technology. And the 3 of those things in combination put us in a very dangerous situation. And so that's really why I'm here today. That's why I do what I do. Now I'm going to transition to going through some research projects that hopefully try to carve out small pieces of that problem so we can better understand how these systems are changing. And actually go in and measure, quantify, and measure again so you see change over time. Try to get a handle on, okay, well, if these systems are out of balance, how can we get them back into balance? So when they are functioning, when ecosystems are working properly, that's what supports us. That's our life support system. So this is, you know, what I call a bird's eye view into the Anthropocene, our time here on the planet. And so this is work from the past several years where we've been doing a comparative study. And these 2 species of birds that I'm going to talk about a little bit are what we find very interesting to compare. One on the left is called the gray catbird. It's a very habitat generalist. It's happy nesting in urban areas and people's backyards and things like that. And then on the right, we have the wood thrush. This is a species that is a habitat specialist, so it's a forest interior obligate. That means it requires intact forest habitat in order to breed and fulfill its life cycle. Yet the 2 are very closely related. They have very similar phylogenies. They're very closely related in evolution and their lineage. They're also very closely related in their life history traits. They're both neotropical migrants, so they overwinter in the tropics. They come up to North America to breed each year. Very similar sort of diets, very similar characteristics. So, but yet we look at that center figure, this is breeding bird survey data. Their population pattern is not showing the same kind of behavior. So why is that? So those start getting my wheels spinning. What sort of questions might come up? Well, why is the wood thrush declining yet the gray catbird seems to be doing okay, even though they're very similar species? So that's what this sort of research aims to try to tease apart. So we look at lots of different metrics to try to compare them. We've looked at survival estimation, so just survival probability, comparing that among these two species in different age and sex cohorts. We look at nest survival, so we go out and do extensive field work where we follow these birds around, we misnet, we capture these birds, we band the individuals, we look at their nests, we can measure fecundity, reproductive success. We look at also the post-fledgling period. Once these birds hatch out of the eggs, they're in the nest for a few weeks and then they come out of the nest. What happens to them? This is a very understudied area in ornithology in general. So we don't know that much. So one way we can try to understand is tracking them and then measure their survival rates by putting little radio transmitting backpacks on them, following around the woods. We could look at sort of their movement patterns on the landscape. So this is sort of habitat use. Once those post-fledgling birds come out of the nest, where do they go? What areas do they use? What areas do they avoid? And how does that affect their survival rates? And then we're also interested in sort of the diet and nutrition. I'm very interested in sort of nutrition limitation. So we know when organisms don't get the proper nutrition or they're either limited, that can reduce their survival, it can reduce how reproductively successful they are. And so we compare how the habitat affects that. This is a cool study we've been doing where we put out these camera traps and these feeding trays underneath different plants. We'll put some trays underneath native plants and some under invasive plants. And we know that these two types of plants, if you have a plant invasion, it's going to actually lower the ability of the wood thrush to find food, which is kind of interesting. So it's trying to connect the dots between plant communities and the insect communities and then the bird communities, how they're responding. And the plant invasion is driven by, guess who? Us, right? We modify those forest fragments, we increase edge effects, seeds come in, plants invade, we bring the plants over from Asia, and so we're the ones monkeying with these ecosystems. And so how are these species going to respond? That's something I'm really fascinated with. So this gets couched in this idea of the importance of small urban forest fragments. That's one area of focus for my dissertation work. And this is just to give you an overview of what I'm talking about here. You know, here's the East Coast, Maine, going down the East Coast. We have this sort of megalopolis situation from Boston down to DC. And we're interested in these small forest fragments, ones that are smaller than 50 hectares. And this is typically about the size of a Best Buy parking lot, one of these box store parking lots. And when you add up all these small forest patches, it actually ends up being a considerable amount of habitat. And who's managing that? What's going on in those small patches? We do know it is breeding habitat for birds, but is it an ecological sink? Is it a trap for these birds to end up going and breeding in? So this research sort of explores that. So this is part of an urban ecology study I'm involved with where we've set up these plots sort of embedded in an urbanized landscape, and we study all these sort of components. We call it the frame, study the forest fragments and managed ecosystems, and we're expanding these sites now. We have folks in North Carolina doing this work with us too, so we're trying to build out this long-term data set where we collect the same types of variables, where we go into a site. It's gridded out nicely. As you can see on the left is an example site web farm. Then we look at everything from the soil chemistry. We sample the invertebrates. There's been tick studies that are done. We collect the leaves, put them in Berlese funnels, and see what sort of insects are in that leaf litter. We look at the understory vegetation community. We also look at vertebrates, mice, birds. And then the overstory, so the dominant tree species. How has that community changed over time? And we sort of are trying to get an understanding of how, when we fragment the landscape, when we modify these ecosystems through plant invasion, through changing land use over time around these areas. How is it going to affect the species that rely on them and breed in them and live in them? And so to highlight that, I'm just going to go quickly through a few studies where we've shown these do have large effects for these species. So this is sort of a comparative study again. We have the wood thrush on the left and the catbird on the right. These are during the post-fledgling phase where they've got little transmitters on them, and we're going to track them around the woods. And just to give you a really brief overview of a bird life cycle, I'm sure most of you are familiar, but they're sort of egg stage for about 10 days to 2 weeks. Then once they hatch out, if they're not depredated at that point, they'll become nestlings for almost 4 weeks. And then once they get to their post-fledgling stage, they'll be there from about 1 to 4 months. And then finally they achieve breeding adult stage, that's recruitment, and they're about 1 year old at that point. So this cycle goes around and around. We want to understand how these different factors affect this transition of the cycle, so we can model that and try to get a handle of which parts are they most vulnerable during. So here's a student collecting telemetry data, and we can sort of plot the different tracks of where these birds go. Each individual is a different color. And so we track these fledgling birds around our Newark landscape. And we took those data points, those location data, and we were able to build these sort of heat maps of where they're using most habitats. So the core areas are the lighter colors, and they're using some of the darker color less so. And this gives us an idea of which habitat they're using and avoiding. We're actually working with a colleague of mine at University of Ghent in Belgium. It's great mathematicians and modelers developed a nice solution, these Brownian bridge models, where you can take any point data and basically create a Brownian bridge, which, if you're familiar with Brownian motion and how particles move in fluid dynamics or in gases, we're using the same idea to create a distribution of where these, in this case, it's the propagules are the animals themselves, how they might move through the landscape. Then what we're going to do is we're going to overlay those bridges, those Brownian bridges, over land cover data or over road data. This is sort of the roads in the area, and we're going to extract the different land cover layers underneath there and try to estimate which areas they're using the most. So again, we're comparing these two species. In gray are the gray catbirds, these gray bars, and in the brown are the wood thrush. And we want to see if they're using different habitats or not. And we actually we subset the data so that there's this pre-independence phase and a post-independence phase. Pre-independence is when the nestling first comes out of the nest. They're actually followed around by the parents for about 20 days. And they're provisioned, the parents are still feeding them, and the nestlings are pretty vulnerable. But then after 20 days, they gain independence. They start getting a little more sort of bold. They explore a little further away from their natal area, and they start exposing themselves to the dangers of what we call the matrix in landscape ecology, areas outside their sort of habitat that they're suited for, and then they encounter things like cats and roads and cars and all these human anthropogenic sort of factors. What we found was pretty interesting, but not surprising, that the wood thrushes are using forest habitat. They're forest specialists, of course. This top bar, they're using it way more than the gray catbirds. And the other takeaway message is that right here, these developed land cover types, that the gray catbirds are going out, they're using this developed land cover more frequently than the wood thrush is. Once we've done that, we actually can compare survival rates as a function of things like number of roads they've crossed. So this is just survival curves for each species. What's interesting is the wood thrush actually has higher survival rates than the gray catbirds during this post-fledgling phase. So that's sort of counterintuitive. If you recall back to that first figure I showed, the wood thrush population was the one going down the toilet. The gray catbirds were actually increasing. So does that make sense that we would find higher survival rates in the wood thrush? It's counterintuitive in a sense. Well, we're only looking at one sort of component of their life history at this point, but this sort of begs the question, okay, well maybe the wood thrush has higher survival during this stage. Maybe it's another part of their life stage that they're experiencing this higher mortality rate, okay? So just sort of interesting that we found this sort of result. And this leads me right into okay, well, what else might be driving these population patterns? What could be the cause? In 2002, this famous paper, Hames et al. These were some heavy-duty ornithologists out of Cornell and Dartmouth. They put out this paper called Adverse Effects of Acid Rain on the Distribution of the Wood Thrush. Okay, so what they did was they did a big analysis, and they did what we call a correlational study. They found that the acid rain being deposited from the atmosphere onto the ground was tightly correlated with the decline in the wood thrush population. So this study was one of these papers that sort of got cited a ton and got into people's consciousness, and everyone started almost saying like, oh, acid rain is causing the decline of the wood thrush, and it was sort of embedded in everyone's thinking, but no one had really tested this yet. So for my PhD dissertation, I said, okay, well, I want to go in and actually test this if these guys, if their sort of hypothesis that these things are connected is even warranted. There's also been some nice studies, some manipulations, experiments from Sarah Pabian and Margaret Brittingham, and at Penn State, they actually went out and they put lime on the soil. They took the soil calcium. So, sorry, let me take a step back. The acid rain, when it comes down, it actually leaches calcium out of the soil. So we have like sulfates, nitrates, things coming that can be produced by coal-fired power plants, things like that. They get up into the atmosphere, it rains down on the ground, and it bonds with calcium, and it washes away, so that calcium is no longer available for the birds. Turns out calcium is like the limiting nutrient for birds. A, because they have to make their eggshells out of it, and nestlings need calcium to develop their bones properly. And it's also really important as a neurotransmitter in general. So that was sort of the thinking Hames et al. And that group was thinking. They're reducing the calcium availability, and this is why these birds are declining. So Sarah Pabian and Margaret Brittingham went out and they actually did an experiment. They put lime down in the soil. And what they did, different species, but a similar species, the ovenbird, the ground nesting warbler in forests, actually, when there was more calcium available, we saw more ovenbirds. The territory density increased. So that was kind of interesting that there was this positive relationship. So we wanted to go in and test this with the wood thrush. This is my question. Are breeding wood thrushes nutrition limited? Particularly, we were focusing on calcium. And so we went in and we collected a lot of data on the food that was available. We surveyed insects that were available there. We looked through all these fecal samples to try and understand, well, what are these birds actually eating, okay? So we had to sample these fecal samples, and then we actually ended up using stable isotopes, another technique where we can trace actual carbon and nitrogen molecules through the food web, and we can trace which foods they're actually eating this way in a more sort of robust manner. And so, yeah, so we got lots of samples from wood thrushes, and we took blood samples to do the stable isotopes. And so then this was sort of the joke question at the beginning of my dissertation work. Do wood thrushes eat snails? Okay, simple question, right? What do you think? Do wood thrushes eat snails? Anyone want to guess yes or no? Yeah, that's okay. Well, we didn't know. No one had demonstrated this in the literature, okay? It turns out it's very hard when you're out there watching birds to see what they're eating, or, these snails can be very tiny. We've done extensive snail surveys, and there are a lot of snails out there, so you'd think they'd be eating them, but are they? We couldn't find them, so actually people were betting. There was actually some wagers that took place in our lab. And then finally, after I'd gone through like 200 fecal samples, I had this moment. And don't worry, I didn't actually jump out of the bath and run naked through our department or anything. But we found a snail, the first one. So I had 100% confidence that one wood thrush ate one snail on occasion. So we're good to go. But we wanted to actually test that in a more robust way, obviously. But the thing is, you can't find these snails. They're so delicate, the shells, they go through the crop and the gizzard of the bird, and it just gets disintegrated, basically. You can't use fecal samples to determine that. So stable isotopes is really the right tool to measure that. So this is basically a mini crash course on stable isotopes, where we measure these sort of heavy versions of carbon and nitrogen that are naturally occurring in the environment. We measure the ratio of those heavy versions to the regular versions, and we measure them in the food sources, all those little types of food they like to eat. And then we actually measure it also in the consumer. So we measure it in the birds themselves. So we took a small blood sample We separate the blood into red blood cells and the white, the plasma, just by spinning it. And then we use mass spectrometry to burn it at a very high temperature and measure the gases that come off that. So that's basically what we're doing in this methodology. And that allows us to track sort of and compare the food and the consumer and then map those two signatures together and figure out what sort of food they're eating. So it actually turned out, this is pretty wild, that they're eating a ton of snails based on our results, okay? You know, about 30% of the diet was snails, and no one would have ever known that from what was in the literature or what you could from looking at traditional food sort of ecology methods. They're also eating these other types of invertebrates, spiders, isopods, earthworms. It turns out that snails, isopods, and myriapods, centipedes, and millipedes are also calcium rich. So we actually also went through and measured the calcium content in all these things to remember to test, are they calcium limited or not? So we went through, so it turns out, a lot of their food is very high in calcium. They're not really calcium limited at all. And so, at least in our system, we can sort of, say that Hames et al., perhaps that's not the correct hypothesis, at least in our neck of the woods where we sampled our birds. Then we did, as an offshoot of that study, we also did another sort of comparison. This is another sort of character I want to introduce, the brown-headed cowbird, into our system, which throws a little wrench into the mix. But this is a species called a brood parasite, like the cuckoos. They've evolved to lay their eggs in other species' nests, and then they actually leave, and they leave the host species to raise their chicks. It's a beautiful co-evolutionary story. Some birders hate cowbirds and are really nasty to them, and so it's actually this beautiful co-evolutionary story. And so what we did was we looked at nests where the nestlings were parasitized and the nestlings that weren't parasitized, and compared their sort of isotopic ratios for carbon and nitrogen. What we saw was that the no cowbirds, these wood thrush up here, had a very different sort of isotopic composition in their tissues compared to the ones that did have cowbirds in the nests. And then we were actually able to show that any of these bars that have the stars over, we found a significant difference between these two, the ones that were parasitized and the ones that weren't. And what we found was that the ones that got parasitized were getting fewer snails, they were getting fewer spiders, which turns out are really important, high in protein and nitrogen, and fewer isopods. Whereas the ones that were getting parasitized received more insects and caterpillars, and these are actually lower quality food items. So potentially, if you get a cowbird in your nest, there's some true reproductive problems with those offspring, because they're getting not as good nutrition to begin with. There's this whole idea, if you don't get nutrition early on in development, that can carry through your entire life, and you can have lifelong sort of reduced fitness consequences because of that. And we also saw that the ones that did have the cowbird, basically they were getting reduced nutrition, and the parents were getting one fewer offspring out of that brood. So there's multiple ways this was negatively affecting the wood thrushes. And this is like another example, potentially why we're seeing that population decline that I presented earlier. So as we moved from sort of what's going on with these individuals and their reproductive sort of limitations, we then wanted to model at the next scale up, the population level, to understand how these populations might be behaving in our urbanized landscape. And so I call this plugging the urban sink. This is the idea of source-sink dynamics. When we talk about meta-populations, we have lots of populations within a big population, and these individuals can move back and forth. A source is a population that produces, it's really good habitat, produces lots of individuals, and those get pushed out into the environment, and they go and then occupy new areas. But a sink is an area that's really probably not the best habitat, it's lower quality, and it just receives immigrants. So there's nothing really produced out of it, it's just sort of receiving individuals. They go into the system, but they're not putting anything back out in the system. So this is this idea of sources and sinks. That's just a little joke in the title about the plan, the urban sink. But anyway, again, this is breeding bird survey data showing you the negative decline in wood thrushes. Areas in red are where they're having, this is changing over time, this delta, we're seeing red areas that are declining. So this whole megalopolis area along the east coast, wood thrushes aren't doing that well. And it turned out my study was also piggybacking on this very long-term data set, over 40, now we're almost 45 years of demographic data, which is unheard of in the bird world. Like, who would study, the same population for 45 years? Like, how can you even do that? Well, a gentleman, Roland Roth at University of Delaware, started with undergraduates sampling these birds every summer with students. They'd go out, they'd follow the nest, they'd band every individual, and they started collecting these data. And so what we're seeing is actually just the number of females in our system, because that's really the important component we focus on in this population model. But we can see sort of, the population isn't doing that great, sort of spikes in the early 90s, then it starts this decline, and this rate is actually right on par with what we see for the regional rate of decline. But yeah, 2,700 birds banded, over 1,700 nests. And so we think this is probably due to this, again, this forest fragmentation and loss. So now I'm showing actually these forest patches that are less than 20 hectares. These birds have very high site fidelity, so they keep coming back every year to the same exact site. And in some cases, that site is no longer there. It could be a new, Walmart or something like that. Or in some cases, it's shrunk so small that they can't effectively breed. So then the whole idea is, how can we reforest these landscapes? This is actually right across the street from my office, this old Chrysler automotive plant. They were building tanks right around the time of World War II. And actually, fun little musical tidbit that actually Bob Marley worked in this factory driving a forklift. That's where the Night Shift song came from. So just a little bit of music trivia for you. But the Chrysler automotive plant was recently shut down and bought by University of Delaware. And so as an urban ecologist, we're thinking, well, how do you revitalize this area? How do you regreen this infrastructure? Now you have this opportunity to actually, create habitat or improve what was once, this sort of horrendous industrial site, and maybe have a new breath of fresh air that can come in there. So it's just getting us thinking about, how do we modify these landscapes to then reclaim that ecosystem function. So here we are. This is sort of the brown-headed cowbird, which you just got a brief introduction to. So here's the nest, which is very typical that we'd find. Blue eggs are the wood thrush, the white speckled are the cowbirds. And some species have evolved the ability to eject these eggs. Like the catbird, for instance, evolved for a longer time with the cowbird.
Does that not cause the mafia?
Oh, it does.
behavior,
It does.
From cowbirds?
Yeah, so he's mentioning something about the cowbird mafia. So these birds, they'll lay these eggs in other birds' nests, but then they watch the nests very carefully. So they sort of patrol their own eggs. And if an egg gets ejected, the cowbird will go up and try to, destroy the host parent's eggs. And there's all sorts of, I've seen lots of crazy activity back and forth. So that's what he's calling the mafia. Yeah, the cowbirds are egg-laying machines. The female will lay like 40 eggs per season, something crazy like that. And so anyway, this is the trajectory in our same site, the proportion of parasitized nests over time. It's something amazing, like 400% increase in the past 40 years. So what we want to do is try to capture all this stuff and build a model so we can understand and then predict into the future and simulate how this population might persist. So the idea was to determine some covariates that are local or regional covariates that might be related to our population rates, like survival, fecundity, things like that. Then we wanted to bake this all into a meta-population model, because we have multiple sites, these discrete forests throughout our study area. We wanted to sort of model that connectivity and movement among patches. And then we wanted to simulate a model. So let's play this fast forward into the future, 30 years, and see what's the fate of our population. And one step beyond that, we wanted to then come up with different scenarios. So what if we modified our landscape in a way that could help, a conservation effort? How would that affect this meta-population? What if we removed cowbirds, for instance? How would that affect our population? What if we actually added forest, or added buffers around the forest patches that exist? How would that influence it? So we were determined to try to model all that. So we took our demographic data, we went out and did more point count surveys to try to estimate occupancy, the probability that a breeding pair of birds would be in any given patch. And we actually collected from those same blood samples that I used for stable isotopes, we could actually do some population genetics as well and look at the relatedness among birds within our patches. And this was sort of our surrogate for movement among patches, this migration and connectivity. So here you can see our patches that we sampled, these sort of 21 sites. All the black polygons, though, are the sites where wood thrush actually occurred. So they didn't occur across all of our patches. And this was actually really important. If they occurred on all of our patches, I wouldn't be able to model the occupancy of these patches because I know all the good ones. I'd have no bad ones to say, okay, well, why are they avoiding these other bad patches, but only preferring these good ones? So we actually needed these patches that were unoccupied to come up with the right models and the right covariates to then say, okay, this is actually what's influencing the probability that we'll find them in any given patch. And so we started out by using these occupancy models from the point count data, and we used an R package called unmarked, where you don't have to mark and recapture individuals. And then we built an integrated population model using these 40 years of data, so very robust. It captures all that annual variation over time that gets incorporated. So we can have high confidence, at least within our system, that the fecundity and the recruitment rates and immigration rates are all being sort of well justified in the model. Did you have a question? Or? Oh, okay, cool. Yeah, if anyone has a question, feel free. You can stop me. We can talk about it. And then we also looked at this, modeling the regression of modeling using regression cowbird parasitism on these covariates. So index of pressure of parasitism. And then finally, this last stage was simulating it. So building a simulation of all these covariates. So real quickly, what we found was that there was this negative relationship of impervious surface to whether or not wood thrushes actually occupied a patch. So this is sort of the model showing you as impervious surface increases, we found fewer and fewer. The probability of finding them went down. And that was actually our top model. We tested a lot of different models and compared them. Then this is from our integrated population model, where you saw our population estimate previously, but now added to that we had adult survival up here and number of individuals that were actually recruited each year. So that's juvenile survival, but it's effectively recruitment. And then we also had fecundity, how many offspring did each female produce per year. And also, because our site is what we think of as a sink, it's really supported by this influx of immigrants each season. So we actually were able to model this immigration rate as well. And then the idea was we could use those sort of demographic parameters and model how it was related to cowbird parasitism. Okay, because we know when a cowbird drops an egg, it's going to reduce fecundity. We know now that the cowbird is actually reducing sort of the nutritional health of those fledglings and nestlings. And so we know there's some negative effects. So the idea was to create these models, and then these are going to be the foundation for our simulation. At each time step, we're going to basically draw from these distributions, okay? We're going to design our scenarios in such a way that we can play with these models to simulate how the system would work. Then the last component was sort of this idea of how birds would move through the system, their transition probabilities. And we use this genetic data to find out who was related to who. If you're more closely related, then that meant there was this connectivity, so there's likely individuals moving between those patches. If they were more distantly related, then there's less likely a movement. So we had a probability. This is what we call a transition matrix. Basically, each cell represents a site, and this is the probability that you stay in the site you're currently in. So as probability goes up toward 100%, that's you can either stay in your site or you can transition to any other site in our system. And that's sort of how those probabilities look. That's sort of a graph version of it, but it's kind of insane to look at it that way. And then this is just another way to conceptualize. Those aren't all the nodes and vertices, obviously, but this is sort of our study area with potential pathways that birds can move among the patches. And then finally, we wanted to simulate this. So we used this sort of a Markov chain approach where we could try to model how these birds would move from one step to the next. And so we came up with these scenarios. The null model was just the current state. We're not going to do anything to our system. We'll just use the data as it is, what we observe. Scenario B was, what if we were able to go in and reforest? Okay, we get permission from the state and municipal state parks to actually, generate forest and plant trees and increase buffers, things like that. How would that influence the metapopulation dynamics? And then finally, scenario C, what if we go in and remove cowbirds? Now this has been really successful, things like Carlin's warbler, black-headed vireo in Texas, where these are critically endangered species, and you have to go in and do something about cowbirds, otherwise the species may go extinct. And it's been really successful. It's very intensive, but again, we're just sort of imagining we have these superpowers to be able to do this stuff. How would that affect the system? Then finally, we combine scenario D, just if we reforest and we remove cowbirds at the same time. So let's take a look, just to go through it one quick time. This scenario, we don't do anything, the null model. In reforesting the landscape, what we want to do is increase the probability of getting wood thrushes in there and breeding. So we're basically reducing this impervious surface and improving that probability of occupancy. The third one, we're trying to jack up survival rates and fecundity and recruitment by removing cowbirds, okay? So that's sort of how we're using these models. So as we reduce parasitism, we get an increase in recruitment or survival. And then finally, we do both of those conservation measures, scenario D. So here's what happened. We simulated that. We did this, thousands and thousands of iterations. Each time step, we sort of modeled this thousands and thousands of times. That's how we got these confidence intervals. And we played it out 30 years into the future. When we just reforested, what we saw was there was an improvement. We saw occupancy increased, and we actually could get our population to persist. It's still not great, it's still declining, but it did something positive, okay? When we remove cowbirds, similarly, at the beginning it's really helping, and then it doesn't quite work as well toward the end of the 30-year period we simulated, but it had a positive effect compared to the null model. And this is what was really interesting. When we combined both, we got this non-additive effect, which blew my mind. But when you go in and you get more birds into those unoccupied patches by improving the habitat quality, and you increase their survival and fecundity, you get this magical sort of effect, okay? So this is a sort of, it's theoretical, it's a model, but this can be used to start to guide actual conservation measures. And the next step would be, well, let's go out and do a manipulation. Let's actually go on the ground and do a little test and see if we can actually, at the site level, improve sort of the fecundity and occupancy of wood thrushes. This is another way to sort of look at those results. Here I'm mapping impervious surface and cowbird parasitism to the growth rate, lambda. So anything above 1 is positive growth rate. What we see here is that it's actually, this impervious surface has a bigger effect compared to cowbirds on lambda. So this is also interesting. Right around 10-15% impervious surface is where they start to have a declining population. That's also where the EPA says watersheds start to become imperiled. That same sort of mark. That's kind of interesting. So that was sort of moving from the individual then to the population. Now I'm going to go very quickly through community level dynamics. So I like to look at these different scales because they each have an important story to tell us. They each have this important, what I call latent information stored within these interactions that can tell us important stuff. So this is work from working with the National Park Service and the National Capital Region Network and the Mid-Atlantic Network. We're actually very fortunate to be involved with this research, helping them design and implement their long-term monitoring programs for breeding forest and grassland birds. So it's a lot of data that we've been involved with collecting and analyzing. And so the idea is we've been collecting these data. I'm actually working with these folks now to improve quality assurance reports. We're building an R package for them right now so they can reproduce these analyses, and so that park managers, biologists can actually see what their data are telling them so they can take management actions. So that's a huge connection. We're trying to close that loop from collecting the data to turning that back into management decisions, and then that adaptive management scheme so we can get that cycle going. These are the sorts of automated reports we're generating for species that occur. We can look at sort of trend analyses on occupancy and abundance. And the idea is with this is an ongoing long-term monitoring program. So we want to continue to revisit these analyses, and we're building the tools to allow them to do that and facilitate that in a nice, easy way. We're also tracking the trend of what we're calling the Bird Community Index. This is an index that's tracking ecological quality. And so the birds respond to changes in vegetation. And this is sort of showing you across different parks. These are all 4-letter codes. They stand for like Antietam National Battlefield Park, Catoctin Mountain Park. I apologize, I'm the only one who understands those 4-letter codes. But anyway, you can see some parks are doing really well, like Prince William has a higher sort of ecological integrity compared to other places like Wolf Trap, okay, which has a much more human-dominated landscape. So this paper we did looked at that. It compared this ecological integrity index in relationship to basically how urbanized an area was. And so what we found was for the National Capital Region Network, the more land cover that was urbanized around parks actually decreases this ecological health and function, even inside the park, okay? But in the Mid-Atlantic network, we found actually there's a positive relationship with forest land cover outside the park, but actually there was no relationship because the Mid-Atlantic is sort of situated in a less urbanized area. So this was sort of an attempt at us looking across an urban to rural gradient and measuring the bird community responses to landscape level urbanization. What we found is that, yes, it's having a negative impact even on some robust parks that we are paying each one of us tax dollars go to supporting these protected areas, and we want to try to quantify that they're actually doing what we intend them to be doing, providing valuable habitat and ecosystem services for us. So very quickly, I know I'm sort of going on with the time a little bit. I'm just going to breeze through some recent exciting projects I'm excited about. I'm also very interested in doing work outside of birds. Birds are a big part of what I do, but this is an interesting project. We're actually modeling the movement of the western corn rootworm through the soil. So who would have thunk, but it turns out I work with a chemical ecologist who's interested in this, and I love individual-based models. So what we're looking at is basically the cross-section of corn roots in the soil. Basically a slice, and these are roots going down into the soil, and they give off these volatile organic compounds into the soil, and that's what the corn rootworm is using to navigate, okay? So we were actually able to model this. It was pretty cool. And some people knew that CO2 is attracting them, and also there's a few different compounds that they've isolated in the lab are attractants, but this is the first time we were able to actually simulate and show, at least in a theoretical sense, that it's this combination of CO2 and the volatile that's really the strongest attractant for the western corn rootworm. This is a new project I'm working on that's been funded where we're trying to model the spread of this invasive insect called the spotted lanternfly throughout the state of Pennsylvania. It's now become of national concern. APHIS has released, I think, $17 million just last year alone to try to control this. We're not going to stop it at this point. It's just sort of controlling spread. So I'm building tools that are trying to identify its host plant, the tree of heaven, using above the canopy imagery. We built a little app that takes Google Street View data and can actually identify the tree of heaven. And the idea is if we can map tree of heaven, then we can actually build a model to predict the spread of this insect throughout the rest of the state. This is just our model sort of in action. This is me just driving down Route 95 in Wilmington, Delaware, and it's picking out every tree of heaven along the road. This is us actually tracking the actual insect itself. So the spotted lanternfly comes in a few different life stages. This is the sort of hatch nymph stage. Then it has another stage it takes place. You'll see in a second. It turns like bright red, has this sort of aposematic coloring. And the idea with this is we want to have the ability to go out and detect these things when they're emerging for the first time. And we want to deploy little cameras out with a chemical attractant a recent paper just discovered which chemicals these guys are attracted to, and we want to deploy this so we can sort of be at the edge of the spread and then collect some other covariates to see which areas there are. And there's the adult. So it doesn't fly until it's the adult stage, but once it's the adult stage, it can fly. It can lay eggs on anything, old wood, logs, your car. So this thing is now of national concern. So we're building this model for the state of Pennsylvania. I just talked to the state of Delaware the other day. They want us to try to build it for them. That's a sort of a cool project. So then the next step would be, okay, we know some sort of tree of heaven distribution. We want to build an individual-based model. So this is just me playing with some models where we know tree of heaven is tightly correlated with roads. It tends to grow in disturbed areas. It will grow along roads. So what I did was I took the Tiger shapefile of all our roads in PA, sort of rasterized that and added a little blur, so taking an average of those roads, and then I added some random noise into the model just to make it a little more realistic. And we can actually model the spread of where spotted lanternfly would go as a function of the roads. So eventually we'll have that vegetation layer, a true distribution map of tree of heaven, but we'll have some sort of similar individual-based model where each time step there's certain probability that it's going to move from the current cell it's established in to a new cell. This is a cool project. If you remember that picture of me holding that like giant funnel in the lab, we're actually collecting rainwater under the forest canopy, something called throughfall, and we're actually trying to extract eDNA from that, so environmental DNA that sloughs off of organisms. And so we're working with this guy, João Setúbal in Brazil. The idea is to eventually sample this along an urban to rural gradient to see how canopy level communities change due to urbanization. So we're doing just the very first pilot study right now where we've got the rainwater collected, we're extracting the DNA using some Kaijin kits and then using a 16S ribosomal RNA marker to see what sort of bacterials the communities exist in the rainwater we found. This is another sort of big data project I've been working on, trying to look at macroecological patterns using, again, bioindicator data of birds, from eBird, from breeding bird survey data, from NextRad radar, also MODIS Terra satellite data. And the idea is to overlay all these data sets and understand how bird communities are changing over time and spatially, why different areas are the communities changing in different ways. So we're using machine learning, TensorFlow to do that. And I just want to end that there are over 400 billion biological indicators that fly around our planet every year. So they can tell us this amazing story if we're just listening, if we know how to get the right data, and we know how to sort of find that signal in all the noise. And that's what I think we ought to be doing. So thank you so much, and I'll be happy to hang out and answer any questions.